Welcome back to Bioclass Bites. In this video, we are going to learn about Mendel's laws of inheritance. Don't forget to subscribe to my channel and like and share this video. In order for us to continue, here are some more terms that we need to learn and we need to be familiar with. So first, genotype. This is part of the DNA sequence or of the genetic makeup of a cell and therefore of an organism. This determines a specific characteristic, so later phenotype, of that cell or organism or individual. So genotypes are part of the DNA sequence. So we cannot see with our naked eyes the genotype. So we assign letters to it. For example, genotype for flower color in this example, we assign the letter P. Okay? Phenotype is the observable characteristic of that genotype or the observable expression of that genotype. So again, genotype, we cannot see it. We assign a letter to it, letter P, for example. Phenotype, this we can see. So we can use words to describe what trait we are seeing. For example, purple flower, that's a phenotype. Or white flower, that's a phenotype. So these are usually descriptions of the organism. Round pod shape, wrinkled seed, something like that. Tall height, or short height, like that. So these are observable characteristics of an individual. Again, genotype, genes, we cannot see it, so we assign a letter to it. Phenotypes, this can be observed with our naked eye. We use words to describe it. For example, purple or white flower. Allele, or the short form of allelomorph, describes variant forms of the genes. Okay? Variant forms of the genotype corresponding to their possible phenotype. So for example, I hope you still remember, alleles can be homozygous or heterozygous. Homozygous, the alleles or the letters that we assign to it, they look the same. Okay? They could be both capital letters. Okay? Capital letter P, so that's a homozygous. This corresponds to a phenotype purple flower. Or it could be heterozygous. Okay? One capital, one small letter. So later, following the law of dominance, even if this is your allele, that still corresponds to a purple phenotype. Another form of allele, homozygous recessive, both are small letters. Homozygous recessive, that corresponds to a white phenotype. Okay, so alleles or forms of a gene can be homozygous, they look the same, both could be capital. Okay, that corresponds to a specific phenotype, or both can be small. So homozygous dominant or homozygous recessive. Or they could be heterozygous, one capital, one small. Now, for future discussion, since we will be using one capital and one small letter of the same letter, sometimes it's best to avoid letters in which the capital and the small letters look the same. One example will be letter P here. Okay, normally we should avoid using letter P because you might get confused and thought that what you wrote was actually a small letter when in fact it should be a capital letter. So letters that the capital and small letters look the same must be avoided, such as C, what else? F, okay, if you have that peculiar way of writing letter F, um, O, P, okay, those words, S, right? Sometimes T can be misleading as well. What else? U, V, W, X. Y can be used if you write it differently. Okay? Z also. Okay? So that those words are usually avoided in reality so that the students will not be confused whenever they write the different alleles of that genotype. Now this is how alleles are used, okay, or how alleles are viewed on how they express the phenotype. Okay, so here, for example, this blue chromosome is what the organism inherited from its father, and this is the red chromosome that is inherited from its mother. Okay, so paternally inherited chromosome, for example, we are looking at this color gene here, the locus or the location for the flower color gene. So for the paternally inherited chromosome, so this is the DNA nucleotide sequence of that allele. So CDA, I hope you still remember this from Central Dogma. We all know that after transcription, translation, and then protein synthesis, this will eventually create a protein. Okay, in this case, it facilitates the production of an enzyme that will eventually show the pigment purple coloration. So for the paternally inherited chromosome, it has this allele, okay? It has this allele, and this sequence creates the enzyme for the purple color. For the maternally inherited chromosome, it has this allele, okay? But in this sequence, this does not, the DNA sequence does not result in the production of the enzyme. Therefore, this DNA sequence results in the absence of the enzyme. So this will not create the enzyme, this particular sequence. However, for the purple phenotype, just having one, okay, having one purple flower allele is enough to actually produce the pigment. So even if this allele does not have the enzyme, the organism still shows the this sequence does not produce the enzyme, the organism still shows the pigment for flower coloration. So this shows us the allele, one allele that produces the trait, then another allele that does not produce the trait. So both of these are present in the cell of that organism. Here are more terms. Character is the heritable feature. Character is the feature. For example, fur color, that's the feature. Trait is the variation of that character. So, fur color, character, trait, brown, black, white, okay? Character could be size, okay? For example, fruit size. Trait is the variation, big, okay, or medium size or small fruit size, okay? True breed refers to having all offspring of the same variety. So when you produce, when all of the offspring produce the same phenotype, okay, after successive fertilization and self-fertilization, then that's considered a true breed. It means that they only produce this trait. For example, this line, it only produces purple flower. Or this line, in this case, for example, it only produces purple flower. Hybridization refers to the crossing of two different true breeds. So when you cross one pure breed with another pure breed, you create a hybrid. For example, pure breed vampire, then you cross it with a pure breed werewolf, then you create a hybrid of a vampire and a werewolf. Just like that. Okay? Then we also label the different generations. P generation refers to the parents. F1 means first filial generation. Okay, so that's the offspring of the P generation. Okay, F2 generation, if this generation had interbreeding, okay, or they fertilize each other, then F2 generation is the offspring of that next generation. Then you can have F3 and then F4 generation. So this is usually avoided in humans because we cannot, for the F generation, all of the offsprings there are siblings. So we cannot reproduce with our siblings. So therefore, we do not have F2 generations in humans. So this one is usually reserved for plants, microscopic organisms, some animals. Okay? So this will just be a review, we've already talked about this. So a cell is said to be homozygous for a particular gene when identical alleles of the gene are present in both homologous of the chromosomes. So Homozygous, homosimilar, it means that it both possess the same allele of that particular gene. Okay, so the cell or organism is called a homozygote. It has homozygous alleles. So there are two types of homozygous configuration. An individual is said to be homozygous dominant when it carries both alleles for the dominant trait. So as you can see, both capital letters, it means homozygous dominant. Homozygous recessive, both are small letters. The cell or the individual is a carrier for both recessive, similar, same, both recessive allele of that particular trait. So both small letters, so or lowercase form is used for that particular allele. Okay, so when you say homozygous, it can be homozygous dominant, both capital letters, or homozygous recessive, both small letters. An organism is said to be heterozygous if it has different alleles of that specific gene. So because of that, it has one capital and one small letter. Okay, so the capital letter represents the dominant allele, and the small letter represents the recessive allele. So if an organism, a heterozygous organism has this allele, okay, and the gene in question follows the law of dominance, even if it is just carrying one dominant allele, the trait, the phenotype or the dominant phenotype will be expressed, while this recessive allele will not be expressed. This will be masked. Okay? So, in a heterozygote, if the dominant, even if there's only one dominant allele present, the trait will be expressed, then this recessive allele will be masked. So I hope you still remember that here, okay? So this is the dominant allele. So that codes for the enzyme to create the purple pigment. Then this is the recessive allele for the white flower. This does not create the enzyme for the pigment. So even if the organism is carrying just one allele for the purple pigment, then the phenotype will be expressed. Okay, that the purple trait will be expressed by the organism. Okay, so again, capital letter and small letter. And the capital letter is usually written first. So capital R, then small r, not small r and then capital R. It's always the capital letter first. Okay? So again, just to clarify, that is for simple or complete dominance. Okay? If there's only two options, then dominant or recessive trait, then in a heterozygous organism, the dominant allele will be expressed. Okay? However, there are some variations in the law of dominance by Mendel. However, that will be tackled in future grade levels, specifically possibly in grade 12. The diagram that we will use to be able to predict an outcome of a possible cross between two parents or breeding experiment is through the Punnett square. And it is named, so this is the Punnett square. This is named after Reginald C. Punnett, who devised the approach. So the Punnett square is a tabular summary of the possible offspring, combination of the offspring, factoring in the alleles of the parents and the alleles of the mother and the alleles of the father. Okay, so this gives us a probability of the offspring of the probability of the genotype of the offspring. So it's a visual representation of Mendelian inheritance, and they could help us predict genotypic and phenotypic ratios of that offspring. Okay, so this is a Punnett square. Okay, so this is how we use the Punnett square. So for when you are crossing just one character or one character, you need to create a 3 by 3 square. Okay, so 1, 2, 3, 1, 2, 3. 3 by 3 table. Okay. On this right side, on this left side, this is where you will write the alleles of the mother. You have to separate, so remember the alleles are always written together. Um, for example, yeah, always written together. But in a Punnett square, you have to separate the alleles per letter. So one letter here and one letter here. This side is for the mother's alleles. Then above on top, okay, this is where you will write the alleles of the father, okay, of the other parent. If you do not know which one is the father or the mother, or do you just write it, just write wherever, okay? But if it is mentioned, the mother has, or the mother has the following alleles, so this is where you will write it, and then the father on top. Okay, then next, then you will cross the alleles of the parents. Okay, as you can see here, number 2, this one is a dominant allele. This one is a recessive allele. So you will just combine them. Okay, write them together. Okay, then you have a heterozygous allele here. Then number 3, then you do that at the bottom. So recessive allele here, recessive allele, then a homozygous recessive allele here. Then number 3 here, dominant recessive allele, then you write it here. However, this one you have to write capital letter first. Okay, not small letter first. It has to be capital letter first. And then last here, dominant, dominant, and then you have a dominant combination. Again, do not follow this part, ha? You have to write capital letter first. Same here, same example here. Dominant, recessive, then you have a heterozygote. Dominant, recessive, another heterozygote. Dominant, recessive, another heterozygote. Dominant, recessive, another heterozygote. So we have another example here. What if you breed this first filial generation? So technically they are siblings, but you can do that in plants. So you just put here the alleles. So heterozygous, one capital letter, small letter, capital, small letter. And then you cross them. So both dominant, then you have um, dominant and the recessive. So this is a heterozygote, then another heterozygote, then both recessive allele. Okay, so that's how you make how you complete a Punnett square. Then after that, you have to count for the phenotypic and genotypic ratios. Okay, we will find that out as we go along. So these are examples of Punnett squares. If you are just crossing one gene, we call that a mono. Mono means one. A monohybrid cross. You are just crossing one letter. Okay? That's just one allele. Okay? If you're crossing two alleles or two genes, we call that a dihybrid cross. So as you can see here, 3 by 3 table for monohybrid, and then 5 by 5 table for dihybrid cross. Two genes. So you know that it's a dihybrid cross because you have two letters, okay? Two genes that you are crossing. And then trihybrid, you are crossing 3 genes. So this will be a 9 by 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. A 9 by 9 table. Okay? And you know that you are crossing 3 genes because you have 3 letters. Okay? So a trihybrid cross is a mating between two individuals with different alleles. So as you can see, there's this variety, okay, of probabilities of the offspring compared to dihybrid and monohybrid. Now imagine how diverse for humans. Okay, we have a total of 20,000 to 50,000 genes. So imagine how many probabilities you can have from that. Now, as we perform the cross in a Punnett square, we will actually put into practice laws of inheritance from Gregor Mendel, namely the law of segregation, law of dominance, and law of independent assortment that we've discussed in the previous video. Okay, and as we go through the discussion, okay, we will further learn more about these laws. The law of segregation states that two alleles for each characteristic segregate during gamete production or during meiosis. So I hope you still remember about meiosis, that the product of meiosis are four haploid daughter cells. Okay, and each of those daughter cells are actually unique because of genetic recombination during crossing over, and that they only possess one copy of that allele. So for example, if this one is a heterozygous pea plant, so you could expect that it has, for example, so pea plant, it has an allele for white and allele for purple for the same gene or for the same flower color. So once this chromosome separates during meiosis, each of the daughter cells, future daughter cells, will get one copy of each gene. So if it has dominant and recessive, one daughter cell will have the dominant allele, and the other daughter cell will have the recessive allele, and then the other daughter cell will have the dominant allele. Okay, same thing that happened here. Heterozygous pea plant. So one recessive allele will go to one daughter cell, and one dominant allele will go to another daughter cell. So this makes the daughter cells unique in terms of combination. So this happens during meiosis. That's the law of segregation. Now, the law of dominance, we've mentioned this a while ago about heterozygotes. It states that if a homozygous dominant trait is crossed with a homozygous recessive, so that produces a heterozygote, the recessive trait will be totally masked. So again, homozygous dominant crossed with a homozygous recessive. So capital T, small letter t, it will create a heterozygote. Okay, all of the offspring are heterozygote, but what do you notice? With their phenotype, all of them are still tall. So even if they are heterozygotes, they are carrying one recessive allele, right? They are still showing that tall trait, okay? The tall variation of the gene, okay? Because one allele is enough to express the trait for heterozygotes. So often the dominant allele codes for a protein, the recessive allele does not. So in heterozygotes, okay, the dominant, all offspring will manifest the dominant phenotype. Okay. So to learn more about those two, law of dominance and law of segregation, I recommend that you watch this video from Teacher's Pet entitled Gregor Mendel. I'll provide the link in the description below. Here are some examples of monohybrid crosses using pea plant's height. Okay, so that's the character. Now, the first thing you have to do is to write down the possible genotypes. So you have here a homozygous dominant and a heterozygous alleles. So they both code for the phenotype tall. And then the homozygous recessive, small letter t, both, it codes for the phenotype short. So that's the first thing you do. And then, for example, you are tasked to make a cross between a pure tall and a pure short, or a homozygous tall or homozygous short pea plant. So after that, following the law of segregation, they will be separated into two. And then, so this is your P, parental generation. Followed by F2, your first filial generation. Now you make the 3 by 3 table. Okay, so you write one set here, capital T. So this is those alleles. So capital T, then here it will go here, small letter t, and then you make the cross. I always write with the capital letter first. So if these are the alleles of the parents, the genotypes of the parents, then these are the genotypes of the offspring. All of them are heterozygotes. Now then, here, you will write the genotype of the offspring. So all of them are heterozygotes. One capital T, one small t. Then going back to your list here, that codes for the, so that's the genotype of all the offspring. Now the phenotype, that codes for tall, height, okay, or tall plant. Then right after that, you're not yet done. After that, you will have to determine the genotypic ratio. How many of the offspring, what's the probability of the offspring to have this genotype? Since all of them have this genotype, you write here 100% heterozygote. Then you also determine the phenotypic ratio. So what's the probability of them having this phenotype? Since there's no other combination, you write here 100%. All of them are tall plants. So if you make a cross between a homozygous dominant and a homozygous recessive pea plant, then you are expected to have all the offspring to be tall. Now let's continue. Imagine if we are Mendel and then we performed either self-fertilization or a crossbreed of these siblings of plants. So now they become the F1 generation. Then their offspring will become F2 or second filial generation. So you just get one trait here, one set of alleles here. So that's a heterozygote crossed with another heterozygote. Then following the laws of segregation, they will be separated. And then you create a 3 by 3 table. Then this will be your second filial generation. Then you copy capital T, small t here. Then you copy it, capital T, small letter t. And then you perform the cross. So both capital, it gives you a homozygous dominant. One capital, one small, it gives you a heterozygote. Same heterozygote. And then both small letter, you see homozygous recessive. So here, for the genotype, you write all the genotypes that you can find in the Punnett square. So you have a homozygous dominant here, two heterozygotes, and then one homozygous recessive. Now these three, going back to your list, they still code, so the phenotype for that, this three still codes for a tall plant, while this one codes for a short plant. So that changes your genotypic ratio and phenotypic ratio. So genotypic ratio, you just count how many are dominant, homozygous dominant. You have one. How many are heterozygotes? You have two. How many are homozygous recessive? You have one. So your genotypic ratio now is 1:2:1, no longer 100%. Your phenotypic ratio, how many are still tall? 1, 2, 3. So you have 3, then 1, 3:1 phenotypic ratio. So 3 tall, 1 short plant. So this is the probability of the offspring. So if you cross the first filial generation, you have 3:1 probability of having a tall offspring. So that's 75%. And one out of 4, so that's 25% probability of having a short plant. Now last sample, what if you cross these two plants? So you just copy it here, heterozygote with homozygous recessive. Laws of segregation, they will be separated, and then you just copy it here. Okay, then separated, then you just copy it here. And then you perform a regular cross. Capital T, small letter t, capital T, small letter t, both of them small letter t, small letter t. Now you only have two genotypes, heterozygote and then homozygote recessive. So this one codes for tall plant, this one codes for short plant. Now, how many have this genotype? 2. How many has this genotype? 2. So 2:2. Then we have to reduce that to its lowest term. So 2:2. So they both have a common denominator of 2. So divided by 2, no? So it gives you 1:1. This is your fine. Always reduces to lowest ratio, lowest term. So 1:1. So that's 50-50. It's true. You can see it. 50-50 chance. Then so that's for the genotype. Phenotype, two two of the offspring are tall, two of the offspring are short. So again, we have to reduce that to 1:1. So this is how you perform the Punnett square, following the law of dominance in this example of pea plant's height. So far, what we are talking about are just characters and traits in pea plants. How about in humans? So these are the observable variable traits in humans. So having cleft chin is a dominant gene over no cleft chin. So look at the mirror and see, do you have cleft chin or do not have a cleft chin? Having widow's peak is dominant over no widow's peak. So you have here that Dracula-like um, structure on your forehead. So that's a widow's peak. Having dimples is a dominant trait. Okay, brown or black hair is actually coded by a dominant gene, and then the recessive is our blonde hair or having blonde hair. Freckles, it's a dominant gene with no freckles. Having brown eyes is a dominant gene, and blue eyes, that's also a recessive gene. So if you notice, no, if a Caucasian, a blonde Caucasian marries a Filipino, you will notice that all of their children have the Filipino traits, which is dark hair and then brown eyes, because those are dominant genes. It's very rare uh, to have an offspring of those parents to have blonde hair or blue eyes, unless they dye their hair or they wear contact lens. Others, for males, having baldness is a dominant trait over or over not having a bald hair. Having abundant body hair is a dominant gene over little body hair. Being tone deaf, okay, is a dominant trait over a recessive normal hearing. Um, Having broad nose, okay, or flat nose is actually dominant over a recessive narrow nose, and broad lips is dominant over thin lips. Now, these are other dominant and recessive traits more. Albinism, okay? Albinism, oh, that's the lack of pigmentation. That's actually a recessive trait. Okay? It's not common. Okay? Cystic fibrosis, that's actually a dreaded disease. Lung failure, liver degeneration, so that's actually a recessive trait. And hemophilia. Other dominant traits, brachydactyly or having short fingers, that's a dominant trait. Hypercholesterolemia, elevated levels of blood cholesterol. That's one actually a dominant trait. Okay, that's why we have a lot of those people with that and more. You can actually pause the video here and read if you want. I recommend that you visit learn.genetics and look at this page on observable human characteristics. You can visit that. I'll provide the link in the description below. Then, the last law of inheritance is the law of independent assortment. According to Mendel, the presence of an allele of one of the genes has no influence over the other. Okay? It states that the inheritance pattern of one trait will not affect the inheritance pattern of another. So it means that I think I've mentioned this, that you inherit the character one at a time. Okay, there's no bulk. Okay, wherein um um characters 1 to 200 you inherit it at once. No, you inherit it one at a time. Okay, so if I think in my analogy, no, you if you happen to have if you happen to inherit a lot from one of your parents, for example, from your father, then it only means that most of your father's traits are dominant. Okay? That's why you have higher chances of inheriting them. But you inherit traits one at a time. You inherit the genes one at a time. They do not influence each other. Okay? So, there. Okay, so I hope you still remember our lesson on meiosis, okay? Wherein the parent cells will produce four four haploid daughter cells that are unique with each other, and then they eventually form a zygote upon fertilization. So, if, for example, if these are sperm cells, and then they happen to fertilize an egg cell, then they will form another diploid zygote. That was thoroughly discussed in our lesson on cell cycle. So you can go back to that if you want to refresh. Now, the reason why we have such variety, I think we've talked about this, about crossing over. So, the reason why we have such varieties is because of this phenomenon that happens during the first meiotic division, during meiosis 1. So, during during prophase 1, or wherein there's a pairing of homologous chromosomes, so they pair, the homologous chromosomes pair up, they tend to have an exchange between the crossing over or exchange of genetic material between two non-sister chromatids. Okay, so they swap segments, they swap parts of their chromosomes and possibly the traits of those chromosomes. And then, so as you can see here, it's pure red. Then you have, of course, this is a simplified illustration. So you have here a pure chromosome and then another chromosome. And then during crossing over, there's an exchange here in the chiasma. So after that, you now have a newly combined chromosome. And then after meiosis 2, they will now produce four uniquely recombinated daughter cells. Okay, they are different from their parent cells. By the way, I don't think I've mentioned this, no? We have a total of 46 chromosomes. That's 23 pairs. One set, we inherited that from our father, and the other set, we inherited that from our mother. Okay? So half of our genome is actually from one of our parents. The other one is from the other parents. That's why if you still remember the lesson on sexual and asexual reproduction, in sexual reproduction, there's genetic recombination of the genetic material of the two parents. Okay, unlike in asexual reproduction, wherein there's no green here, all red, because you only have one parent which donates or which gives the genetic material. But since we reproduce through sexual reproduction, half of our genetic material came from our father, the other half came from our mother, and then after that it will produce um, recombinated sex cells after meiosis 1 and 2. Now, crossing over again is very important because it allows genetic recombination. It allows genetic diversity. Imagine no two daughter cells are the same. They are different and unique from each other. So if these are egg cells, each one is a unique egg cell. If these are sperm cells, each sperm cell is a unique sperm cell. They carry that unique combination of um, genes. Okay, so this is important because this leads to variation among offspring, which increases the likelihood of them to survive. And if you still remember, we did that experiment on paramecium reproduction, and we have determined that sometimes it's not really, it does not work out if you just have only one type of only one type of genetic material. Okay, so I hope you remember that. So these are more illustrations of crossing over. So this one, during meiosis, prophase 1, the pairing up of homologous chromosomes, and then in the chiasma, that's the site of crossing over, there's an exchange of genetic material, and then after anaphase, after meiosis 1, you see we have now unique recombinated chromosomes. So and then following meiosis 2, then this is the product of meiosis 2. very unique recombinated daughter cells. This is a comparison between meiosis with and without crossing over. So if there's no crossing over, they just separate like that. So, okay, there's no chiasma here. At the end, you just have two types of combination of the daughter cells. So imagine this will be the sperm cells or this will be the egg cells. While if there's crossing over, there's the chiasma here. Okay, there's the exchange of genetic material, and then following meiosis 2, as you can see, there are 1, 2, 3, 4, four unique, four different types or unique daughter cells. So that's why crossing over is important because it provides, it processes genetic recombination, all to ensure survival in the environment. Now, this is the monohybrid cross problem set A. By the way, for this video, we will just be doing a monohybrid cross, okay? So you can pause the video now and then you can try to solve the following problems, okay? Sige. Now let's see, I'll show some of the solutions, okay? So let's try number 1. So it says here that determine the genotypes, phenotype, genotypic ratio, and phenotypic ratio of a purple flower, a heterozygote purple flower with a homozygous white flower. So you write here homozygote, a heterozygote crossed with a homozygous recessive. So capital R, they will separate, small letter R, then you copy it here, then you write it here, copy it here, and then you perform the cross. Capital letter R, small letter R, capital letter R, small letter R, and then both of them, you cross them, both are small letters. So that gives you the following genotypes, heterozygous and homozygous recessive. Now this codes for a purple flower, this one codes for a white flower. So that gives you 2:2, 2:2, okay? And then we have to do the lowest form of that. So that gives us 1:1 genotype. Then the phenotype, 2 are purple flowers. Then 2 of the offspring they have white flowers. So 2:2, or we lower that into 1:1. Now you can pause the video here so that you can see the solutions to some of the problems. You can study them on your own and see and compare. So try to answer them first and then compare your answers with this. Okay, you can pause the video here. Then finally, this one, a problem set. Okay, so a paragraph problem set. So you can pause the video and try to determine the answer. Now, Mendel's laws are not observed all the time. The world is not divided into tall and short people. We have people in the middle. The world is not divided into two traits. We have a lot of mixing and hybridization. So these are examples of non-Mendelian patterns of inheritance. It means that they do not follow Mendel's laws of dominance. For example, polygenic inheritance, it means that many. More than one gene can affect a single trait. For example, I've mentioned height, okay, and eye color. Pleiotropy, a single gene can affect more than one trait. For example, diseases such as cystic fibrosis and sickle cell anemia. Okay? Multiple alleles. Genes can have more than 2 alleles. Okay, so the genes can have more than 2 forms. For example, the ABO blood typing in humans, so blood type A, blood type B. Incomplete dominance, for example, there could be an intermediate trait. For example, instead of red and white, you have pink, for example, in Japanese 4 o'clock flowers. Or codominance, no single allele is dominant. For example, human blood groups. Environmental factors, the genes may be affected by the environment, such as the Siamese cats, wherein the enzyme is activated by the temperature. Okay? And gene interaction, products of a gene can interact to alter genetic ratios. Okay, for example, production of purple pigment in corn and coat colors in mammals. This one we will not discuss anymore for grade 8, but when you go to grade 12 under genetics, this will be thoroughly discussed in that level. But if you are interested, you can actually go to the online library and check out the grade 12 General Biology Genetics lecture PDF wherein all of this will be discussed. Okay, you can access the link in the PDF or you can just proceed there in the online library. Now, so what we've discussed are mostly just the foundation of genetics, but one of the applications of genetics will be genetic engineering, particularly creating recombinant DNA. Um, and right now, we are using recombinant DNA to edit our genes and to cure diseases. So this video will tell you more about that. So I recommend you watch this. So how gene editing is curing disease. I'll provide the link in the description below. I hope you notice here the nucleotide bases from our lesson in Central Dogma in Molecular Biology. Then for your reflective journal logs, still talking about the application of genetics in genetic engineering. So genetic engineering will change everything forever through CRISPR. Okay, this one is for Corixast. I recommend that you watch this video and I'll provide the link in the description below. That ends our very long video. I hope you learned something new. Don't forget to subscribe to my channel and like and share this video. Till next time, goodbye.